In 1964, two Australian scientists gave a name to a smell humans had recognized long before laboratories could explain it. Isabel Joy Baer and Richard Grenfell Thomas called it petrichor, the distinctive odor released when rain reaches dry earth and stone. But the strangest part of the phenomenon can happen before the first drop lands where you are standing. A dry road can still be untouched, while the air suddenly turns earthy, mineral, almost electric, and unmistakably like rain. For generations, people treated that sensation as intuition, folklore, or a mysterious ability to sense approaching weather. Then chemistry began separating the smell into molecules, and one of them proved almost absurdly powerful to the human nose. Its name is geosmin, a volatile compound produced by microorganisms in soil, especially bacteria in the genus Streptomyces. Humans can detect extraordinarily small amounts of it, concentrations low enough to make contaminated water taste earthy before it looks unusual. That sensitivity once inspired an irresistible evolutionary story. Perhaps our ancestors smelled geosmin to locate water across dry landscapes. But the evidence does not prove that humans evolved a dedicated rainfall detector, and geosmin is not normally released before moisture touches soil. The real explanation is more interesting, because rain can reach your nose before the storm reaches your body. Rain falling kilometers away can aerosolize earthy compounds, and moving air can carry that chemical signal ahead of the advancing shower. Thunderstorms can add another warning molecule, ozone, transported downward and forward by turbulent winds before precipitation arrives. And in 2024, researchers identified a human olfactory receptor that responds selectively to geosmin, a protein called OR11A1. For the first time, the famous smell of wet earth could be connected to a specific molecular sensor encoded in the human genome. Yet even that discovery did not prove the survival story people wanted it to prove. It opened a deeper mystery about why our nervous system notices one ancient bacterial molecule with such remarkable persistence. The trail begins not in clouds, but underground, inside organisms that were producing the smell long before humans existed. Streptomyces are filamentous soil bacteria, famous for an entirely different reason. Members of this group have given medicine many antibiotics, but in the soil they live complex lives, growing branching networks and eventually producing spores. During that developmental cycle, some species release geosmin, the molecule whose name literally evokes the smell of earth. Geosmin is a terpene built from carbon and hydrogen with an oxygen-containing alcohol group. Its structure is compact, volatile enough to enter the air, and extraordinarily recognizable to many noses. It contributes to the earthy flavor of beets and can become a serious nuisance when cyanobacteria or actinomycetes release it into reservoirs, aquaculture systems, wine, fish, or drinking water. Baer and Thomas reached the idea of petrichor by studying rocks and clays that released a characteristic odor after moistening. They proposed that during dry periods, materials derived partly from vegetation could accumulate on mineral surfaces and then be liberated when water returned. Their work predates the modern high-speed imaging of raindrop aerosols, but it captured the same essential transformation. Dry ground stores chemistry, wetting mobilizes it, and the atmosphere suddenly acquires a smell that was effectively waiting on the surface. Geosmin later became the molecule most closely associated with the earthy note, yet petrichor remained a broader sensory mixture. That distinction matters whenever someone says, the nose is detecting rain. The nose is actually detecting volatile compounds whose abundance and transport are changed by weather. The concentration required to make geosmin noticeable is tiny, although the exact threshold depends on whether it is measured in water or air, the experimental method, the individual, and which form of the molecule is tested. In a 1992 sensory study, people were substantially more sensitive to naturally occurring negative geosmin than to its mirror image positive form. That detail matters because smell receptors operate through molecular fit. Two compounds can contain the same atoms arranged as mirror images, yet interact differently with the proteins lining the nose. The olfactory system is not measuring earthiness as an abstract quality. It is detecting molecular structures and translating patterns of receptor activity into perception. Humans possess roughly 400 functional olfactory receptor genes, plus many receptor pseudogenes left behind by evolution. Each functional gene can encode a receptor protein expressed by subsets of sensory neurons high inside the nasal cavity. Odor molecules bind these receptors in combinations, beginning electrical signals that eventually become recognizable smells inside the brain. For decades, geosmin posed a surprisingly specific problem. Scientists knew humans were highly sensitive to it, yet they did not know which human receptor was doing the detecting. Insects offered clues. Fruit flies have a highly specialized pathway for geosmin and often avoid it, demonstrating that evolution can assign enormous behavioral importance to a single earthy molecule. Humans, however, do not possess the same insect receptor. Our olfactory system evolved through a different receptor family. Researchers therefore had to test human receptors directly. And that search finally produced an answer published in 2024 by a team working with the Technical University of Munich and collaborating institutions. They screened a library representing 386 human odorant receptor types, along with hundreds of common variants. In that experimental system, one receptor stood out. OR11A1 responded to geosmin, while the other receptors in the screen did not cross the researchers' response threshold. The result provided the strongest molecular link yet between human DNA and the smell of wet earth. The receptor discovery also exposed an important limit in the word specific. In the cell library used by the researchers, OR11A1 was the only human receptor that crossed their response criterion for geosmin under those conditions. That does not mean every molecule in petrichor is routed through OR11A1, or that geosmin can activate nothing else in living tissue. It means the receptor became the clearest, experimentally demonstrated human molecular sensor for the compound. The brain never receives a label reading geosmin. It receives a pattern. Activity across glomeruli is processed by networks leading toward olfactory cortex and regions deeply involved with emotion and memory. Context completes the perception. The same earthy signal can be interpreted as a garden after rain, a muddy riverbank, a beet on a dinner plate, or an unpleasant taste in drinking water, because perception is chemistry filtered through experience. This also explains why measuring odor sensitivity is difficult. A detection threshold is not a universal constant like the boiling point of pure water. It depends on delivery method, concentration steps, airflow, temperature, background odors, adaptation, participant age, attention, and the statistical rule used to define detection. Claims that every human can sense exactly 5 parts per trillion compress a range of experiments into a number that sounds more certain than the evidence. What remains exceptional is not one internet-friendly threshold, but the consistency with which extremely small geosmin signals become perceptible. That makes the receptor system useful, even if evolution never selected it specifically for storm prediction. A molecule associated with soil microbes can provide early information about water, food quality, microbial growth, or environmental change. In nature, one chemical clue can carry several meanings at once. OR11A1 is encoded by a gene in the human genome, translated into a G protein-coupled receptor capable of recognizing chemical information. When geosmin engages the receptor, it can initiate the signaling cascade that an olfactory neuron uses to tell the brain that a relevant molecule has entered the nose. That is the genetic connection hidden inside the title. But it is not a simple rain gene. A receptor detects a molecule, not a weather forecast. Geosmin can appear in water, food, soil, plants, microbial cultures, or humid air without any rainstorm nearby. The brain interprets context by combining OR11A1-driven input with hundreds of other chemical signals, visual information, temperature, humidity, memory, and expectation. Even more intriguingly, the 2024 researchers examined related OR11A1 receptors from several mammals. The response to geosmin was conserved across the species they tested. A version from a desert-dwelling kangaroo rat proved far more sensitive in their cell-based assay than the human receptor. That cross-species conservation suggests geosmin has long carried ecological information worth detecting. What information? For the bacteria making geosmin, the answer became clearer in 2020. Researchers studying streptomyces found that geosmin and another earthy compound, 2-methylisoborneol, attract tiny soil arthropods called springtails. The attraction was not accidental background chemistry. Production of the volatiles was connected to the bacteria's sporulation program. Springtails approached the odor, fed on the bacterial colonies, and picked up streptomyces spores on their bodies. Spores also passed through the animals and were dispersed in fecal pellets. The smell of earth was acting as a biological message. To the springtail, it advertised a resource. To the bacterium, it recruited transportation. That discovery completely changes the emotional meaning of petrichor. The smell humans associate with cleansing rain and fresh landscapes may originate partly in chemical communication between microorganisms and animals. When you smell geosmin, you're intercepting a signal produced inside an ecological network far older than cities, agriculture, or our species. Rain then becomes the delivery system. In 2015, researchers at the Massachusetts Institute of Technology used high-speed cameras to examine what happens when droplets strike porous surfaces. They found that raindrop-like impacts can trap tiny bubbles at the interface between water and soil. Those bubbles rise and burst, ejecting microscopic aerosols into the air. The process resembles an almost invisible spray generator. A drop hits a porous patch. Air trapped beneath the spreading water forms bubbles. The bubbles reach the surface and burst. Tiny droplets are launched upward, carrying material from the ground into the atmosphere. Wind can then transport those aerosolized compounds beyond the location where the rain actually fell. This mechanism helps explain why the smell seems to leap from the earth after a light shower. Heavy rain is not always better at producing the effect. The efficiency of bubble formation and aerosol release depends on impact speed, surface properties, moisture, and other conditions. Moderate impacts on suitable porous surfaces can produce particularly strong aerosolization. 2 years later, follow-up experiments showed that the same raindrop mechanism could aerosolize viable soil bacteria. The discovery was larger than smell. It meant rainfall could physically move microorganisms from soil into air through microdroplets. A familiar sensory event was connected to environmental microbiology and the atmospheric movement of living material. Geosmin and other volatile compounds can ride within this burst of airborne chemistry. Once lofted, they do not remain directly above the wet patch. Air currents disperse them. If a storm is moving toward you, rain may already be striking fields, soil, vegetation, or roads upwind while your location remains dry. The molecules arriving at your nose therefore become chemical evidence of rainfall that has happened somewhere nearby, not necessarily rainfall that has already happened to you. This is one of the simplest explanations for the apparently impossible claim that a person can smell rain before the first drop hits their skin. You are not smelling the future, you are smelling the storm's leading chemistry. A gust front can outrun the rain shaft. Turbulent air beneath and around a storm can move odor-bearing aerosols horizontally. If petrichor has already been generated beneath the advancing cloud, the wind can deliver it first. By the time you recognize the earthy signal, the precipitation that released it may still be beyond the horizon, hidden by buildings, terrain, or darkness. Thunderstorms introduce another molecule capable of arriving early. Ozone consists of 3 oxygen atoms rather than the 2 found in ordinary molecular oxygen. It has a sharp, pungent odor sometimes compared with chlorine or the smell near electrical equipment. Atmospheric electrical processes and photochemistry can contribute to ozone, and storm circulation can transport ozone-rich air downward. Powerful downdrafts can pull air from higher in the atmosphere toward the surface and spread it outward ahead of precipitation. A sensitive person may notice that sharp chemical smell as the storm approaches. It is different from the warm, earthy character of geosmin, yet in experience, the two can merge into a single impression. Rain is coming. That means the phrase, smell of rain, hides multiple phenomena. Petrichor itself is not one pure chemical. Baer and Thomas originally described an odor associated with materials released from dry rocks and soils after wetting. Plant-derived oils and many other volatile compounds can contribute. Geosmin supplies the famous earthy musty note, but the complete atmosphere after rain is chemically complicated. Cities create their own version. Rain falling on asphalt and concrete can release accumulated oils, dust, organic residues, vegetation-derived molecules, pollution products, and other compounds. A person raised in a dense city may associate rain with a different mixture than someone raised beside farmland, desert soil, forest litter, or tropical vegetation. Memory then amplifies the chemistry. Olfaction has unusually direct connections with brain regions involved in emotion and memory. Odors can evoke places and experiences with a speed that feels almost involuntary. The first smell after a dry spell may therefore seem stronger than its concentration alone would predict because the brain immediately attaches the molecule to storms, childhood, heat, soil, or relief. None of this requires a mystical weather sense. It requires a very good chemical detector paired with a brain trained to recognize patterns. If every time a particular mixture of ozone, geosmin, humidity, cool air, and wind is followed by rain, the nervous system learns the association. Eventually, the odor becomes predictive even though each molecule obeys ordinary physics. The genetic component enters at the level of detection. Olfactory receptors are proteins encoded by genes, and human receptor genes are highly variable. 2 people can carry different versions of receptors that respond with different strengths to the same odorant. Genetic studies across many smells have shown that receptor variation can change perceived intensity, pleasantness, or even whether a molecule is detectable at all. That principle is well established for odors such as androstenone, cilantro-associated compounds, floral molecules, and musks. A single receptor variant can sometimes produce a large perceptual shift. In other cases, many receptors and many genes contribute. Smell is a combinatorial code, so genetic influence does not imply that every odor has one simple gene determining sensitivity. For geosmin, OR11A1 provides a specific receptor target, but the human genetics of natural variation in geosmin perception is not yet mapped well enough to say that people who smell rain earlier carry a particular proven OR11A1 allele. That claim would jump ahead of the evidence. The receptor has been identified. The population-level genetic story remains incomplete. There is another puzzle. The concentration of geosmin required to activate human OR11A1 strongly in laboratory cells is much higher than some psychophysical estimates of human detection thresholds. That mismatch does not invalidate the receptor. Cell assays are artificial environments, and receptors behave differently inside living olfactory tissue, where proteins, signaling amplification, mucus, binding partners, and neural processing alter sensitivity. It may also mean the sensory system extracts weak signals through mechanisms that a dish cannot reproduce. Thousands of neurons expressing receptors can amplify information. The brain can integrate repeated molecular encounters across time. Additional interactions may remain undiscovered. OR11A1 is an important piece, but the nose is more than one receptor floating in isolation. This scientific uncertainty matters because the evolutionary claim is even less settled. Humans absolutely depend on water, and in arid landscapes, the ability to recognize cues associated with moisture would be useful. Geosmin can signal wet soil, microbial activity, standing water, or ecological conditions connected to moisture. It is tempting to imagine natural selection sharpening our sensitivity for precisely that reason. But temptation is not evidence. No study has demonstrated that ancestral humans carrying a particular geosmin receptor variant survived drought more often because they could smell distant rainfall. We do not possess fossilized olfactory thresholds from early Homo, and we cannot reconstruct the exact selective pressures that shaped OR11A1 across millions of years. The receptor's conservation across mammals suggests biological relevance, but different species can use the same molecule for different purposes. One animal may approach it, another may avoid it. A third may use it as one cue among many. Even within the same species, the meaning of a smell can change with hunger, reproductive state, previous exposure, or environmental context. Fruit flies offer a dramatic example. Geosmin activates a dedicated pathway that drives avoidance because the odor can indicate microorganisms associated with unsuitable food and egg-laying environments. Mosquitoes and other insects may respond differently depending on species and context. Springtails are attracted because Streptomyces effectively recruits them into its reproductive cycle. Humans often describe geosmin as pleasant when it appears faintly in petrichor, but unpleasant when it contaminates drinking water or food. The molecule is the same. Concentration and context change its meaning. An earthy hint after summer heat can feel refreshing. The identical sensory quality in a glass of tap water can signal that something is wrong. Water utilities know this problem well. Geosmin is generally regarded as an aesthetic water quality issue at the concentrations where people notice it, not automatically as evidence that the water is dangerous. Yet customers may detect it before laboratory instruments reveal any other obvious change, precisely because human olfaction can be extremely sensitive to earthy, musty compounds. This sensitivity has fueled the popular claim that humans smell geosmin more effectively than sharks smell blood. That comparison is usually misleading. Detection in air cannot be directly compared with chemical sensing in water by taking two concentration numbers and declaring one species thousands of times better. Different media, molecules, receptors, experimental methods, and behavioral thresholds make the arithmetic scientifically weak. The remarkable fact needs no shark. Humans can detect geosmin at very low concentrations. In the 1992 work on geosmin enantiomers, sensitivity differed sharply between the naturally occurring form and its mirror image, showing that the nose was responding to molecular geometry with precision rather than simply reacting to a generic earthy chemical class. That precision is what DNA makes possible. The OR11A1 gene provides instructions for a receptor whose three-dimensional shape allows interaction with geosmin. Mutations changing receptor structure can, in principle, alter that interaction, just as variants in other olfactory receptor genes change smell perception. Evolution works on these molecular differences generation after generation, but hard-coded does not mean immutable. The brain learns odors. Age can reduce olfactory sensitivity. Viral infections can damage smell. Smoking, medications, nasal disease, pollution exposure, hormones, attention, and expectation can all change perception. Even if two people carry identical receptor sequences, they may not consciously experience petrichor in exactly the same way. The environment also determines whether the signal exists at all. Dry soil can accumulate plant oils and microbial products, but without the right moisture and impact conditions, little may become airborne. A light shower after a long dry period can produce an intense smell, while days of continuous rain may produce far less dramatic petrichor because surfaces have already been washed and saturated. This explains the emotional power of the first rain. The dry interval stores chemical potential. Volatile residues accumulate on mineral and plant surfaces. Soil microbes pass through their own cycles. Then the initial droplets strike, creating a sudden aerosol burst. The contrast between dry background air and the new odor plume makes the change unmistakable. Wind performs the final trick. Your nose has no access to the ground kilometers away, but molecules do not care about boundaries. Once airborne, they travel with moving air. A storm approaching across open terrain can therefore announce itself chemically before its rain shaft crosses your position. The phenomenon feels like prediction because your sensory system receives information from an event that is spatially separated from you. Sound does something similar with distant thunder. Light from lightning arrives before the sound. Smell is simply slower, carried by air rather than electromagnetic radiation. But it can still report conditions occurring somewhere else. In that sense, smelling rain before it falls is real. What is wrong is the idea that the nose senses moisture before moisture reaches any soil. Usually, something has already happened upstream. Rain has struck ground, storm circulation has shifted atmospheric chemistry, or environmental humidity and wind have released or transported odors toward you. The ancestral value of noticing those signals remains plausible. Water availability shaped every stage of human evolution. People navigating landscapes needed to find rivers, springs, vegetation, animal trails, damp ground, clouds, and storms. Smell was one source among many. A faint earthy plume could have contributed information, especially when visibility was poor. Yet there is no reason to assume rainfall detection was the only selective pressure. Olfactory receptors evolve under enormous ecological complexity. Geosmin can indicate microbial contamination as easily as fresh water. Detecting it could influence food choice, drinking behavior, habitat assessment, or avoidance of spoiled resources. The same receptor may have served several functions across evolutionary time. The 2024 receptor study itself emphasized geosmin's role as a semiochemical and suggested that OR11A1 could function as a warning system in food selection. That interpretation is important. Humans may be sensitive not because geosmin means safe water, but because earthy microbial signals can carry information about the condition of food and water, sometimes attractive and sometimes undesirable. The kangaroo rat receptor adds another layer without solving the mystery. Its unusually high sensitivity in the laboratory fits an animal whose ecology makes chemical information about soil especially relevant. But even there, receptor sensitivity alone does not reveal behavior. An animal must integrate smell with motivation, memory, and environmental context before the signal becomes action. This is why evolution rarely produces the simple stories we prefer. There is a gene, there's a receptor, there's a molecule, there's a behavior. But the arrows connecting them are not always one-directional. One receptor can respond to more than one ligand. One odor can activate multiple pathways. One behavior can arise from many sensory cues. What makes geosmin unusual is how much of that chain is now visible. We know microorganisms synthesize it. We know it participates in ecological communication. We know rain can launch soil-derived material into the air. We know people detect geosmin at extremely low levels. And now we know a human receptor capable of recognizing it. The mystery has moved. It is no longer, how can anyone smell rain? Chemistry can explain that. The unanswered question is why this particular bacterial molecule occupies such a sensitive place in mammalian olfaction, and what selective history maintained that sensitivity over deep time. Perhaps water-seeking contributed. Perhaps food and water quality mattered more. Perhaps geosmin was simply one reliable signal in a complex landscape where microbial activity, moisture, soil, vegetation, and animal behavior constantly overlapped. Evolution does not need a molecule to mean one thing forever. It only needs detection to improve decisions often enough to matter. That possibility fits the way humans actually experience petrichor. The smell does not force a single behavior. It captures attention. We notice it. We turn toward it. We recognize a change in the landscape. The sensory system is delivering information first. Culture and memory decide whether the response becomes relief, caution, nostalgia, or anticipation. And somewhere inside the nose, OR11A1 is part of that ancient transaction. A volatile molecule released from bacterial chemistry encounters a receptor built from genetic instructions carried in human cells. The receptor changes state. Neurons fire, signals move toward the olfactory bulb, and the brain reconstructs an environment that may be kilometers beyond sight. Then the first drop finally lands.